Hereby I open this academic ceremony in which Xiaodi Zhang will defend the academic thesis, Interplay of methylglyoxal and immune cells, implications for type 2 diabetes. May I invite you to present a summary of your study and the conclusions of your thesis.
Yes, thank you. Dear Prorector, Corona members, supervisors, colleagues, family, and friends, thank you so much for coming here today for my PhD defense. I would like to take the next 15 minutes to present an overview of my research entitled Interplay of methylglyoxal and immune cells, Implications for Type 2 Diabetes. People with type 2 diabetes are at high risk of developing cardiovascular disease. Treatments for type 2 diabetes can reduce its burden. However, a residual risk of cardiovascular disease remains in these people with type 2 diabetes. And inflammation is thought to play a key role in this residual risk of cardiovascular disease. Under inflammatory conditions, immune cells in our body are activated, and within these activated cells, such as inflammatory macrophages, glycolysis is elevated. Glycolysis is a process that breaks down glucose to produce energy, and this process can further lead to an increase of inflammation in these activated macrophages. Methylglyoxal, in short MGO, is a dicarbonyl compound. MGO is mainly formed as a byproduct of glycolysis. MGO is highly reactive. It binds to proteins to form advanced glycation end products, AGEs. AGEs are something that we don't like in our body. They are harmful to our cells. In type 2 diabetes, an elevation of MGO levels and AGE levels are found in people in human plasma. And moreover, excess accumulation of MGO and AGEs are also found in inflamed tissues and are linked to cardiovascular disease. Increasing evidence has shown a link between immune cells and MGO. However, how exactly they interact remains unclear. So my thesis mainly focuses on investigating whether and how MGO is formed from glucose, especially in immune cells, and how excess MGO affects immune cells. So the first part of my thesis is about formation of methylglyoxal. To investigate whether methylglyoxal formation is derived from glucose intake, in humans, we give them labeled glucose, and we measured labeled MGO in the plasma. And we found MGO formation in the plasma is 100% derived from glucose intake. And this increase in MGO formation further leads to an increase of MGO-derived AGEs in plasma. In mice, we performed the same experiment, so the mice also received labeled glucose, and we found again MGO formation in the plasma is completely derived from glucose intake. And this is also true for tissues and also for immune cells. Moreover, we were also able to perform the same experiment in a mouse model with type 2 diabetes. And we found the glucose intake derived MGO formation is further increased in plasma, in tissues, and in immune cells. Next, we also investigated whether glycolysis contributed to MGO formation that derived from glucose intake. So again, we performed the same experiment in mice, and we blocked glycolysis. We would expect to see that MGO formation is also blocked. However, we didn't observe a reduction of MGO formation in the plasma or in the immune cells. We only found that glycolysis contributed to MGO formation in the liver, in skeletal muscle, in brown fat tissue, and in bone marrow. So MGO formation is derived from glucose intake. However, not all MGO formation is dependent on glycolysis. Apart from the MGO formation, to our surprise, we found that MGO concentrations are very high in immune cells. In humans, MGO concentrations in immune cells are 20,000 times higher than in plasma. And in mice, similarly, MGO concentrations in immune cells are about 800 times higher than in plasma. And these findings made us wonder whether MGO would have an effect on immune cells. So the second part of my thesis is about effects of methylglyoxal on immune cells. Talking about immune cells, here I would like to focus on monocytes and macrophages because they are the key players in the inflammation. Macrophages are derived from monocytes, and human monocytes can be divided into three types: non-classical monocytes, intermediate monocytes, and classical monocytes. And the intermediate monocytes are especially linked to the clinical outcomes of cardiovascular disease. And also, the macrophages accumulation are always observed in the atherosclerotic plaques and contribute to cardiovascular disease. And to investigate how MGO affects immune cells, we have performed a lot of experiments, both in vitro and in vivo. In vitro, we added MGO directly to the cells, to macrophage and monocytes, and we didn't observe a direct effect of MGO on these cells, on inflammation. And in vivo, we injected mice with MGO, and again, we didn't see a direct effect of MGO on inflammation in mice, and also no direct effect on monocyte or macrophage recruitment in mice. However, when we collected cells from these mice that received MGO injection and then cultured these cells for a week, then we observe an enhanced inflammatory response in these cells. And these findings reminded us of trained immunity. And what is trained immunity? Trained immunity happens in innate immune cells. Normally, these immune cells don't have a memory, but they can be trained under certain stimulation, and the training basically based on the DNA modifications and is accompanied by an elevation of glycolysis. And once these cells are trained, they can respond higher to a second stimulation. And to confirm our idea that MGO is involved in trained immunity, we trained human monocytes. And during the training, we found that MGO formation is increased. And when we blocked MGO, we found that the enhanced inflammatory response, is also blocked. Therefore, we confirmed that MGO is involved in the induction of trained immunity, and this is possibly via DNA modifications. So the effect of MGO on immune cells is not directly on inflammation or directly affect immune cell recruitment, but it induces trained immunity. And to connect our experimental work to the clinical data, we further analyzed the data from the Maastricht study. The Maastricht study is an observational prospective population-based cohort study in 10,000 participants and with an oversampling in individuals with type 2 diabetes. And with a subset of this data, we studied the associations between fasting levels of plasma MGO and circulating immune cell counts or activation. And we found that fasting MGO levels are associated with higher numbers of intermediate monocytes and non-classical monocytes, and with lower activation of intermediate monocytes. And since intermediate monocytes are linked to cardiovascular disease, our results support that the interaction between intermediate monocytes and MGO may explain the high risk of cardiovascular disease in people with type 2 diabetes. In conclusion, our thesis supports an interaction between immune cells and MGO. Immune cells produce MGO via glucose metabolism, and MGO doesn't directly affect immune cell counts or activation, but MGO induces trained immunity in immune cells. And under type 2 diabetic conditions, the interaction between immune cells and MGO can be a vicious cycle and can further contribute to the development of cardiovascular disease. Thank you so much.
Thank you for your presentation. The opposition will be opened by Professor Koenen, Professor Biochemistry of Vascular Inflammation and Thrombosis at this university. Professor Koenen was the chair of the assessment committee and he is the secretary of the degree committee.
Thank you, Mr. Prorector. Dear candidate, before I ask the questions, I would really like to congratulate you with this very interesting work. A lot of consistent and well-performed studies and also very well written. And I had a hard time finding mysteries in your data, but I found a couple and I would like to ask your help to unravel them. The first is related to the pie diagram that you show on page 77 in figure 4.1. And you also showed it in your presentation, which was very clear, that there's a 20,000-fold higher concentration of MGO in the cells. And that results that you only have quite a low concentration of MGO circulating in plasma of only 3%. If I look at this data, and I also look at your chapter 7, where you use plasma MGO as a marker, my question is, are you measuring the right pool of MGO in the blood? Which pool of MGO in the blood would you measure with the knowledge that you have now? Would you change it, or would you, please?
Highly esteemed opponent, thank you for your question. Well, based on our findings, I would, in future study, I would definitely study MGO in the immune cells. Mm-hmm. However, that doesn't really mean that MGO in the plasma is not important. yeah, MGO is produced normally in cells, but in the plasma, it also can reflect the MGO from tissues and from the cells. It reflects different things from the intracellular MGO.
Okay,
Yeah.
so you say there's a higher level of information coming also from other tissues
Yes.
in the plasma MGO.
Yeah.
Okay, that's a good answer for a start. I'm already satisfied with that. But then I would like to draw your attention to page 80, figure 4.4. And I just would like to discuss a little bit about the contributions of the relatives. So you have measured with 13C MGO glucose, you measured 13C MGO production. You see that the cells respond quite well, but relatively is only 2% or 0.5% contribution in the white blood cells. In plasma, it's 30 to 40% dependent on external glucose. So that means that your plasma, although you get more information, is also subject to more variation, which you may not want to have in larger studies. At least that's my opinion. Maybe you would look differently. And also about pre-analytical variables. Suppose you take blood in the Maastricht study and cells get activated. What would happen with the plasma MGO levels then? And how can you ease a little bit my concern that pre-analytical variation and also donor-to-donor variation are not influencing your data, for example, in chapter 7?
Yes, in the blood MGO, if the immune cells are activated, MGO can be produced more and can be released into plasma. But based on my knowledge, not in the clinical, if people do an OGTT, they normally do several tests before the time zero. So
Okay.
when it's steady, then we use the blood samples.
Okay.
Yeah.
So you mean you already see not much variation at the baseline, and then you know that the blood, that the glucose and the MGO that you see is actually from the metabolism and not from broken cells.
Yes, indeed.
Okay, good. Then I'll take that as your answer, and then I will go to page 59 because there is the mystery I already alluded to a little bit. And maybe you can help me as the expert to unravel it. I would like to draw your attention to panels E and F. So in E and F, you use BSA, and you incubated it with 2 different concentrations of MGO. And then you see a decline in MGO over time, which, in my opinion, would be the reaction of MGO with BSA, because MGO alone, you don't see a decline.
Yes.
So there's no... And then if you look at the half-lives of MGO, it's both 2 hours. So meaning at 100 micromolars and 10 micromolars, you have the same half-life of decline. That would mean that the reactions go equally fast under these 2 conditions.
Yes.
Okay, you agree. Very good. Then in panel C, you look at the modification. So at the reaction product of the reaction, that's a modified amino acid, I think, on BSA. And then suddenly you see that with 10 micromolars, you get much less modification of BSA than with 100. So my question, and that's the mystery, is what happened with all the MGO that it looked like reacted in panel E and F, but in panel C, you see a big difference. And if you don't know the answer directly, I don't mind that much. I can give you 2 options that I think might be happening.
Yes.
Okay. The worst option is there is a contamination in BSA that captures away your MGO. And then after this contamination has fully reacted, then you see the decline you see with 100, or the rise. That would be the worst. And the other one might be that you're looking at a modification of BSA, but there might be a different modification of BSA you haven't looked at. Could that be an option?
Well, the first one, I think that's not a problem, that BSA is contaminated.
Mm-hmm.
And the second one, a different modification.
Different modification in BSA, and you're looking, of course, at only one amino. You look at two modifications. One doesn't apply. That's a CED. I think it's CED, and the other one looks like it applies, but it doesn't. It doesn't explain the full decline of the MGO. So.
Yeah, that is possible.
Okay, good. Then let's stop here. With regard of the time, I give the word back to the pro-rector. Thank you for your answers.
Thank you.
Thank you. The opposition will be continued by Dr. Bito, Associate Professor Cardiovascular Physiology at Hasselt University in Belgium. Dr. Bito was also member of the assessment committee, and we highly appreciate your participation to this academic ceremony.
Thank you very much, Pro-Rector. I would like to start by first, I'm sorry, there's a lot of echo. So I want to first congratulate the candidate for the work performed during her PhD, for the quality of the manuscript, but also for the presentation of today. I don't see anyone anymore. I hope you can hear me. Yeah, you hear me, right? Can you hear me?
Excellent,
yeah.
Excellent.
Okay. Sorry. So I enjoyed, just to finish with the congratulations, I enjoyed reading your manuscript and I learned a lot. So thank you for that. I have a few questions for you, but my questions are more general. The first one is about MGO and immune cells and maybe their contribution in autoimmune diseases. I was wondering, what are the molecular mechanisms by which MGO interacts with the immune cells? Are there specific receptors or signaling pathways involved in the interaction between MGO and immune cells? And can potentially MGO-induced changes in immune cells contribute in one way or another one in the development or autoimmune diseases? What is your opinion on that?
Yeah, in autoimmune disease, yeah, I'm not an expert in autoimmune disease, but based on our findings, I think MGO, because we found that MGO has an effect on inducing trained immunity. So MGO produced in the immune cells, and it is possibly modifies DNA in the cells, and then that leads to a training of the immune cells, and this modification is can be in NF-kappa B pathway, and that may contribute to the autoimmune disease, in my opinion.
Okay, so via training immunity then?
Yes.
Okay, no, I'm not an expert either. I was just wondering because there's a big relation between your immune cells, the immune cells and MGO. So I was wondering if there was a potential link. So that's fine for me. I have another question regarding MGO and diabetic inflammation. So what is the relation between MGO levels and the severity of the inflammation in diabetes? Because I was a bit puzzled in chapter 6. You mentioned that you have an increased MGO in the plasma and that is related to low inflammation levels, right? So I was wondering, is there a direct relationship or not between MGO levels and the severity of the inflammation in diabetes? Well, I will open my book. It was page 124. There you're right. Higher MGO concentrations in plasma are, but they're associated with less low-grade inflammation. And then I was a bit puzzled. What's now the answer? High MGO levels are related to high inflammation, or is that the other way around, or how does that work?
I'm sorry, I haven't found the sentence.
Oh, it's page 124. Oh, wait. It's really at the end. It's in agreement with his findings. We recently found in a study in humans that long-term higher intake of dietary MGO is associated with higher MGO concentrations in plasma, but with less low-grade inflammation. And that's where I was a bit confused, because I thought high levels of MGO were associated with high-grade inflammation. So maybe you can clarify that for me.
Yes, long-term higher intake of dietary MGO is associated with higher MGO concentrations in plasma.
Maybe I can simplify my question. Are high levels of MGO associated with high levels of inflammation, or is that directly related to inflammatory status in type 2 diabetes, or it's not always the case?
MGO is, it's linked with higher MGO. Yes, dietary MGO, long-term injection of dietary MGO can lead to higher MGO concentrations in the plasma that is found in the previous study, and that's related with less low-grade inflammation. I would think that is because the immune cells are at exposure to MGO in long term, and then the immune cells get exhausted, and therefore with less low-grade inflammation.
Okay, thank you for your answer. That's fine for me. That was an open question, to be clear. And maybe a last question, if I still have a bit of time. So the levels of plasma MGO reflect the levels of MGO in the tissues, right? So I wonder whether MGO could be used to monitor the treatment or the treatment response of specific therapeutic efficacy in diseases where inflammation plays an important role. Could we, if we monitor MGO, can we actually use it to monitor the efficacy of certain handling, certain medications given to patients who have type 2 diabetes or who have another disease where inflammation plays an important role? What's the clinical translation of your findings? That's my question.
Yes. We think MGO can be a target of the metabolic disease, and if we target MGO, so lower MGO concentrations in the disease, less that can help reduce the burden of the disease, because then there will be less stress from MGO on the cells, on the tissue. So I would think that targeting MGO can benefit the metabolic disease.
Okay, thank you very much.
Thank you.
Thank you. The opposition will be continued by Professor Savelkoel, Professor Emeritus Cell Biology and Immunology at the Wageningen University. Professor Savelkoel was member of the assessment committee, and also for you, we highly appreciate your presence during this academic ceremony.
Thank you very much, Mr. Prorector. Respected candidate, I also would like to congratulate you on the completion of your thesis, and I also would like to extend my compliments to the supervisors, the promoter and the co-promoter. I read your work with great interest, but I still have a number of questions that I would like to discuss with you. So one of the things is that in your thesis you put MGO really at the center. Now, there are many different types of ages that can be derived from glycated proteins. And my question is, did you consider to look also at other types of glycation, and might they be compensating for some of the effects that you might observe with one particular type of ages, namely MGO? Can you speculate on that?
Highly esteemed opponent, thank you for your question. If I understand correctly, you want me to explain other types of glycation on
so if you from
MGO?
Yes, from glycolysis you also might let's say obtain CML modification of proteins, which also leads to ages.
Yes.
And they might be given a different type of profile of particular types of ages, and they might interfere with different types of receptors, and therefore have a different type of response. And since these profiles are so heterogeneous, some of them might compensate for some effects, others might enlarge the effect of MGO. Did you consider this?
Yes, MGO can react with proteins and produce Ages, such as MGH1, CEL. And Ages have several receptors, such as RAGE, receptor of age, and HR1, R2, or 3. And it combines to the receptor of ages and then contribute to the inflammation and disease.
Okay. So there are different types that you might consider in your work, and different consequences of that. Now, you also said that not all MGO comes from glucose, and so, for example, gut microbiota can also produce MGOs, and therefore I was a bit surprised that in your mouse studies you analyzed a number of different organs that you also presented in your presentation, but did you not consider to study also the intestine on these mice, or maybe even the microbiota itself, but at least the intestinal tissue?
Yeah, in the future study we can definitely study MGO in the intestine. Of course, some MGO can be produced from microbiota, and but most of MGO is still from glucose, based on the current knowledge. More than 90% of MGO is from glucose.
Okay, thank you very much for that. Now, then in chapter 6, you studied this trained immunity that you also mentioned a number of times already before. Now, that could explain, let's say, why besides having direct effects of MGO on the cells, you might also change the responsiveness of these cells, and therefore you incubate them with MGO in this setup for studying trained innate immunity. There are quite a number of questions to be discussed there, but in view of time, just a few. One thing that you see, and that has been published before, is that you see increase in TNF production, but no effect on IL-6. And you say, yeah, I see training because I see more TNF, so this contributes to inflammation, etc. You did not really discuss this in depth. Do you have an explanation why you see this difference in both these pro-inflammatory cytokines? Because normally speaking, they would go up both at the same time.
Yes, in this chapter, we reported that the enhanced inflammation is basically based on a significant increase in TNF, but not significant in IL-6. But in my opinion, we can still see a pattern of increase of IL-6, but that is maybe the inflammation is not strong enough. To see both of them that are very significant. And also here we only did experiment based on the samples from 3 participants. So I would think if we include more participants, if we can have more power.
Yeah, I think also you yourself gave a nice other explanation, and that's the fact that you also wanted to look at NF-kappa B activation, which is primarily involved in TNF signal transduction. But for IL-6, it's much more complicated. There are different signaling pathways and that may interfere with each other. So I would definitely urge you to study also NF-kappa B activation in these cells.
Yes.
Yeah. And then you mentioned also in your presentation different types of macrophages. Classical type, non-classical type, intermediate, etc. But did you also consider to look more at, let's say, different differentiation stages of these macrophages, more M1-like pro-inflammatory activation linked to infectious diseases versus M2, which is a much more immunoregulatory phenotype of macrophages? There are different cell surface markers that you could study.
Yes. Yeah, this is more for future study.
Okay.
Yeah, for our current study, we haven't looked into the differentiated macrophages, but only the regular non-activated macrophages.
It would also give you a nice opportunity to study epigenetic changes that you mentioned, but you did not analyze them yet. Yeah. But I would definitely advise you to do so.
Yeah, that's for my future research. Yeah,
thank you very much for your answer. Then I return the word back to the pro-rector.
Thank you. The opposition will be continued by Professor de Galland, Professor Internal Medicine, in particular in diabetology at the Maastricht University. Professor de Galland was also member of the assessment committee.
Thank you very much, Mr. Pro-Rector. Dear candidate, I first like to start with congratulating you with achieving this thesis, and also like to extend my congratulations to your supervising team. I enjoyed reading your thesis very much. It teaches me a lot, where you looked at the interplay between glucose, methylglyoxal, and inflammation. And one of the things that you teach me is that quite a few dietary products contain MGO. One of them is coffee and some dairy products. Now I start my day very often with coffee. Now, given the relation between MGO and diabetes and cardiovascular disease, should I stop this habit? And should we advise people to stop drinking coffee to reduce the risk of diabetes? What do you think?
Yes, well, coffee contains a lot of MGO, and we in our group, although I didn't really study the dietary MGO in real life, but in our group, we have other projects, and they have studied that MGO in coffee and also in other food. In dietary intake of MGO, if you don't inject too much, like more than 3 cups of coffee, is not really that harmful.
Okay, thank you very much for that answer. So I can continue my habit of drinking 1 cup of coffee today. Thank you. Okay, so let's move to the inflammation then. So I would like to focus on chapter 7, where you used the Maastricht study. You also mentioned in the presentation you studied MGO fasting and after a glucose load in people with type 2 diabetes, those with pre-diabetes, and people with normal glucose metabolism. And what you find is that there is a relation between fasting MGO and the number of intermediate monocytes and non-classical monocytes. But post-glucose load, you don't find such an association, right? And then in the interaction analysis, you find there is a relation between fasting MGO levels and classical monocytes, people with type 2 diabetes, but you see actually lower levels of classical monocytes in those without diabetes. So can you first tell me the difference between all these kind of subsets? I mean, in terms of inflammatory capacity between these monocytes and also in relation to cardiovascular disease?
The different type of monocytes, based on the literature, is intermediate monocytes and non-classical monocytes have been reported multiple times that they are really linked to cardiovascular disease, but not much are reported based on classical monocytes.
So, and what's the difference in inflammatory capacity between these three groups?
Yeah, sorry, I didn't study this before.
So what I know of it is, I mean, the non-classical are more pro-inflammatory, and the classical are more immune modulatory. So that would fit in with having a relation with cardiovascular disease. I think that is also what your suggestion is based on our study on this chapter, right? Yeah. Now. I'm a bit puzzled about the post-glucose load, because I think that postprandial glucose is more related to cardiovascular disease in type 2 diabetes. Now, you don't see this relation with MGO, right? So does MGO, I mean, in relation with inflammatory cells, then really has a link with cardiovascular disease in this people?
Yeah, in our study, we didn't find an association between postprandial MGO levels and immune cell counts or activation. I think an important reason is that for this study, we only have the data from fasting immune cells. So we only collected the immune cells at the fasting state, but not at postprandial state, and I would think that make this link not that obvious.
Right. So
Yes.
what would you do if you could have done it again? I mean, if you would have if you would do a new study.
Yeah. If the data set is available, I would definitely study both the postprandial state MGO levels and at postprandial state. Yeah, the
right.
Immune cells.
Okay. Now, one more question, if I may, which is a very short one. So, you also see a kind of downregulation of the function of intermediate monocytes in relation to fasting glucose levels, right? And you then say this is a kind of an exhaustion because of the repeated inflammatory stimulation. What kind of repeated inflammatory stimulation are you talking about? I mean, what's the repetition there?
Sorry, you mean the exhausted immune cells are related to?
So you say that these exhaustion is the consequence of repetitive inflammatory stimulation, and I was just wondering what kind of repetition in inflammation you are referring to. Because what I would think of is that what is repetitive is hyperglycemic events after meals, for example. But then again, you don't see that in your study. So it's all about fasting. So I think it's more chronic stimulation rather than a repetition.
Yes. Yeah. The exhaustion is mostly based on the long-term exposure to higher levels of MGO, and that leads to an exhaustion in immune cells.
Right.
Yeah.
Do you think that's protective against cardiovascular disease risk?
Yes, and I would think that can contribute to the development of cardiovascular disease.
Well, I was thinking about the reverse. Do you think it's protective against cardiovascular disease? If it's
protective?
Yeah, in terms of it's exhausted because the function is not as good anymore.
No, I think the exhausted cells, yeah, maybe one part is a kind of protection to the cells. But on the other hand, if the cells are exhausted, they don't function normally, so they don't react very well to further stimulations. So that I don't think that's a good sign for the disease. If, for instance, if people with type 2 diabetes, if they get infection, and the immune cells don't react very well, yeah, then
okay,
yeah,
in terms
it's worse.
Of disease. Okay, I have a number of questions, but my time is up, so I give the word back to the prorector. Thank you very much for the answers.
Thank you. The opposition will be continued by Dr. Wheaton, Associate Professor Transplantation and Translational Immunology at this university, and Dr. Wheaton was also member of the assessment committee.
Thank you. Respected candidate, also I want to congratulate you with a beautiful thesis, and I enjoyed your very clear presentation a lot. And of course, my compliments are also for your supervisors. And one of the things that I really liked in your thesis is the fact that you use different models, mouse models, in vitro models, and also human samples. So I think that really contributes to the translational capacity of your data. So well done. I like to challenge your conclusions in chapter 6 a bit, if I may. I think it's a very interesting and intriguing chapter where you conclude that MGO has no direct effects no very obvious direct effects on immune cell counts, activation, but you do describe a role in trained immunity. Right?
Yes.
I actually want to start with a very simple question. You already explained it. What is your definition of trained immunity?
Esteemed opponent, thank you for your question. Trained immunity is, to make it simple, is a long-term memory in innate immune cells.
Mm-hmm. Okay.
Yes.
Yes, I fully agree. What I learned from your thesis, of course, and from your presentation is that for trained immunity, you have a stimulus, you get a response in the immune cells, it goes down, and then a second stimulus will give a bigger response, right?
Yes. Yeah.
In my opinion, that resting phase is a hallmark of trained immunity as compared to priming, where you also activate immune cells, but they don't really go down, even though the secondary response may be amplified. How in your model, in your chapter 6 experimental setup, can you discriminate priming versus true training?
In my study, we first give MGO, and we don't directly see an enhanced inflammation. So I would think that's the first stimulation, and afterwards it goes down. And then after a few days of resting and a second stimulation, and then we do see an enhanced response. So I would think our study is related more to trained immunity than priming effect, because we don't really see the response directly from the first stimulation with MGO.
Yeah, and why did you then choose a different setup for your figure 6.1, where you look at the direct response, and if I see it correctly, you stimulate, you give 1 time 25 microgram IV injection, and in your experiments in figure 6.4, where you look at trained immunity, you inject 1 milligram. Is that correct?
Yes.
Why did you choose a different setup and not do it in one experiment?
Yes, in figure 6.1, it's based on 25 micrograms of MgO. That's where we start. We started to use 25 micrograms of MgO to inject mice, and we didn't see a direct response. And at first, we didn't think this is trained immunity, so we tried a different concentration of MgO. So we tried also this 1 milligram. And later we cultured the bone marrow cells from these mice. Yes, both from 25 micrograms and from 1 milligram, and we saw that 1 milligram MgO injection led to a higher response of inflammation after the LPS stimulation on immune cells. So we present this.
And did you do in one experiment the 1 milligram where you also looked at the direct effects of the 1 milligram?
Yes, yes,
okay.
We also look at that.
And there was no effect?
No.
Okay.
No consistent effect.
Okay, good. Then I want to move a bit to a more conceptual question. You show that if you inject MgO, that your bone marrow cells that get sort of primed and they respond better to LPS. And in your human experiment, you use beta-glucan-induced trained immunity, and there if you inhibit MgO, the effect of beta-glucan training disappears. Do you consider MgO more as a trainer, or is it more an amplifier of the training effect of perhaps other trainers like beta-glucan?
Yes, I would think MgO in itself maybe not strong enough like the beta-glucan to induce trained immunity, but it also because we also have a condition that only with MgO on human immune cells that can induce, it can also by itself induce trained immunity to some extent, but not that strong compared to beta-glucan.
And could, if you think in your discussion and in your valorization, you also say that maybe in healthy individuals that trained immunity is primarily beneficial, but if it's too much or if you have a combination of factors, it may become detrimental. Do you see a role for MgO in that as well? That it can basically change the balance in the immune system?
I think that if you give a shock of MgO to healthy people, that may also have a bad effect. For instance, induce trained immunity, but you don't see that directly. So you don't see it directly on your blood, on white blood cells, etc. But it can be a potential risk. And if you later get some challenge like infection or something, maybe then you respond worse than the other. That without this shock of MgO, but normally, like I just mentioned, in like the dietary intake of MgO, that's I think that's not a problem.
Okay, thank you very much.
Thank you.
Thank you. The opposition will be continued by Dr. Donners, Associate Professor Vascular Inflammation and Disease at this university.
Thank you. Dear candidate, I obviously also want to congratulate you with this very nice piece of work and also your promotion team. I also really enjoyed it, and I'm particularly intrigued also by this trained immunity concept, and you already had some discussions about it. You mentioned that it's kind of a memory. It involves DNA modifications, epigenetic changes, glycolysis. I was wondering, is this trained immunity only leading to an enhanced response to a secondary stimulus, or can it also be the other way around, that it dampens the response?
As deemed the moment, thank you for your question. Based on the definition of trained immunity, upon the second stimulation, it is always enhanced inflammatory response. And if it's opposite, then we call it tolerance.
Tolerance.
Yes.
And that's exactly where I want to go. So I was wondering about the mechanism. So you mentioned the DNA modifications and glycolysis for the trained immunity. How about the mechanisms behind tolerance? Is that also involving similar things, or is it a different mechanism?
If I remember correctly, that is also relevant to the DNA modifications.
Mm-hmm. Okay, so in that sense, also a little bit of a trained immunity, but with a different outcome then.
Yeah.
Okay. Then I would like to proceed with one of your propositions on trained immunity, proposition number 6. Maybe one of the paronyms will like to read it.
The potential involvement of trained immunity is hyperkalemic, memory complicated, the research for therapeutic modulation to target inflammation.
Yes, thank you. So a proposition with different aspects in it. I would like to ask you to elaborate a little bit on what you mean. So what do you think, how does trained immunity complicate the research for therapeutic targeting of inflammation? Can you elaborate?
Yes, because if MGO can induce trained immunity, so that is the first stimulation, and then for trained immunity, the second stimulation can be from a different organism. So then once the cells are trained by MGO, then it can another stimulation, like from bacteria or other stimulation, can it's not once the cells are trained by MGO, they don't. Even if you lower MGO concentrations in the future, that cannot really reduce or eliminate trained immunity. So they still respond to. They still can have a harmful effect, and this is the long-term memory on innate immune cells.
So what you would like to say is that we should focus with our therapeutics on this, in principle, this second hit that the immune cells encounter.
Yes, but not only for the therapeutic, but also maybe for the daily habit. We don't ingest too much MGO. So then we stop it from the beginning. Mm-hmm. We don't induce this trained immunity.
Yeah.
Yes.
And can you target the trained immunity itself? So are these modifications in the DNA, are they reversible? And would that be a potential target then?
I think for now, more and more research are about this therapeutic on trained immunity, and I think that can be possible in the future.
Okay.
Yeah.
Xiaodi Zhang, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Xiaodi Zhang. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Schalkwijk is authorized to confirm upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of significant integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Xiaodi Zhang, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee affixed with the official seal of the university. Congratulations, Dr. Xiaodi Zhang. Very well done. And but of course, Christian will pronounce the laudatio as the most important and first supervisor. Christian, word is yours.
Dear Xiaodi, Dr. Zhang. Now that sounds amazing, doesn't it? Yes. You did it. Getting a PhD is the highest academic degree that you can get, and it's the result of hard and challenging work, very challenging at times. And doing it in another country, in another language, that makes it even more impressive, in my opinion. I checked it in my email box, and it all started in 2017, somewhere October, when you reached out to Casper and me, stating that you would like to do a PhD, and then we talked via Skype, I think, or Zoom, Skype probably, back then. And you left a very good impression to us. One thing that I do remember of our talk back then is that you asked how far Utrecht was from Maastricht, because your boyfriend back then lived in Utrecht, and you were happy to know that it was so short a distance, only 2 hours by train. So it makes you think how relative things are. But anyway, we got your CV, you got your reference letter, and both of them confirmed our impressions. And I actually reread your reference letter from your previous supervisor, and I was wondering, is it all true, or was it all true? So I checked out some of the statements of your previous supervisor, and I checked whether they were true. So one of them was that Xiaodi is a quick learner and able to do experiments independently. And I think it turned out definitely to be true. You learned quite fast about quite complex things like flow cytometry, primary human samples, and the MBO probe. And that needed a lot of optimizing and very tight planning of your time, because the samples were coming in every 15 to 30 minutes during the OT-GTT study participants. But I think you managed this all very well. Also, very close collaboration with the other members of the team, so that was really nice. The second thing that was mentioned is that you were good at doing animal experiments in mice. And after arriving in the Netherlands, you soon learned that doing animal experiments in the Netherlands, it's not so easy, right? It's not so straightforward. And especially for you, it was extra hard, but I remember you had to learn from this big book, this additional entrance exam on anatomy. And then you had to write amendments to the ethical approvals, working protocols. It was really not so easy. And then in the end, then you started doing your first experiments. And I also remember that the first test day, about everything went wrong that could go wrong, but it showed that you're a fast learner. And after a while, you were harvesting different organs, making antibody mixes. I could not follow what you were doing, and I just had to do what you told me. That was really great. Xiaodi showed some great skills in management and led and helped undergraduate students. And I think, indeed, although during your first years it seemed that you were maybe a bit hiding after the technicians in the lab and in lab management, you really grew in your independency, especially after the retirement of José. You started also to successfully supervising undergraduate students who are now also continuing their scientific career, so that's also partially your merit. And along the way, at the end, you steadily actually became a boss in the lab. You had a good overview of what was going on, and you also sometimes confronted me if I was maybe expecting too much from master students, so it was really nice having you in the lab. One of the other things was she has a rigorous scientific attitude, and she never forged data in any experiment. This sounds like very logic thing, but it's very important. What I always liked is that you were very open when things did not really go as expected. And boy, things went not as expected a lot of times. I cannot even start to count how many times we incubated cells with MGO, and we measured things, we injected mice with different concentrations, and each time that we thought we found something, we failed to reproduce it, and you repeated experiments, you made extensive data overviews, performed literature searches, and in the end, we had to conclude that MGO just did not affect immune cells, at least not in the way that we thought they did. But your rigorous way of performing those experiments and honest brainstorms about failed experiments, they were really instrumental in advancing our knowledge on the importance of MGO, and also the importance of the experimental conditions in which you would measure them. You also found that this bizarre high concentration in immune cells, and although it landed on a little bit of skepticism, and you remeasured, recalculated, it just turned out to be right. So that will definitely advance the field, and it shows also how important it is, everything, that we know how things do not work. I only have 2 traits left. I won't take too much time. She takes everything very seriously, was in the letter, and I think that's also very true, and maybe a little bit too much. Because I remember when COVID started, we wanted to write this review during the lockdown, but that advanced very slowly because I got only really small pieces of text, and it shows that you really want to be serious about giving high quality work. But it also showed that you had that quality in you, and in the end, it just went on faster and faster, and the thesis was in the end finished more or less on time. Right? This is also true with respect to your presentations. You put a lot of effort in making really nice slides. You integrate feedback very well, and your presentations are so nice and well prepared that even I can give them if I need to take over. So kudos to that. And finally, I think that the most important thing that was in your reference letter is that Xiaodi has good relationship with others. She's optimistic, positive, and never complaining. And I think that was also, and I think I speak for everybody, that is true. You're very nice and kind to be around, socially evolved with all the lab members. I also remember that the first week you arrived, we had a strong beer together at the bar. We drank a Belgian duvel when you came to watch one of my games. So yeah, it was clear from the start that we would get along just fine. Although it does seem that you might be a little bit more careful with white wine, but other than that, it's fine. So, it turns out that everything your previous supervisor wrote about you is completely right, and I'm happy we could work together on this not always so straightforward project. And as I mentioned, although you started out perhaps a little bit shy, not at full speed, you grew remarkably along the way to become a true scientific researcher. I also mentioned it. I often call you my qPCR, with an exponential increase at the end. And now you continue your research in the lab of Emile van der Vorst, where you were selected of 50 candidates to start there. So that made me honestly very proud, just as you did today. Congratulations.
Thank you.
Dear Dr. Zhang, also on behalf of the Maastricht University and the Board of Deans, I congratulate you with the degree you have acquired. It is already mentioned you will continue in the MGO research at Aachen University, and the university wish you all the best in your future academic career. Hereby, I will close this ceremony. Before congratulating the candidate, we will make a picture here in the aula in the presence of Professor Bito from Hasselt University, and afterwards we will take a picture with a new doctor on the stairs in the central hall. Hereby, I close this academic ceremony.